With this method, we can accurately determine the number of mutations in a single hematopoietic stem cell. These mutations can be used to decipher mutational processes and dissect hematopoietic lineages. Our method avoids the use of whole genome amplification techniques by clonally expanding hematopoietic stem cells in vitro, which results in lower error rates and benefits downstream analysis.
The mutational patterns present in the genomes of hematopoietic stem cells can reveal the mutagenic and DNA repair processes to which they were exposed.
Begin this procedure with preparation of sample material and staining as described in the text protocol. Then prepare 25 milliliters of hematopoietic stem and progenitor cell or HSPC culture medium. Fill a 384 well cell culture plate with 75 microliters of HSPC culture medium in each well. To prevent evaporation of the medium in the outer wells, fill the outer wells with 75 microliters of sterile water or PBS and do not use these wells for cell sorting. Correct sorting of viable signal HSPCs is the most critical step in this protocol. Set gates for the HSPC sorting based on an unstained control in 10,000 cells from the stained sample. Gate single cells by drawing a gate around the linear forward scatter height versus forward scatter area fraction. Use the unstained control fraction to draw a gate for the lineage fraction. Draw gates for CD34 positive cells and further characterize this subset by setting a specific gate for CD38 negative CD45 RA negative cells. Load the 384 well plate on the fax machine and sort single cells. If applicable to the